Today we're going to get into invertebrates, the features that we use to classify them, their characteristics, and all the many diverse phyla within the invertebrates. There's a lot of material we're going to cover. We'll probably break this up into several different lectures, but for now, let's just jump right in. So first, we need to explore some definitions and characteristics of our invertebrates. Firstly, invertebrates are simply animals that do not have a backbone, and they are very morphologically diverse, as we'll learn throughout this lecture. We do use different key features of their anatomy and physiology to classify them. Some of these include their body plan, the presence or absence of a head, the number of germ layers that they have during embryonic development, and then how does this organism digest its food? Specifically, what type of body cavity or gut does it have? And where is the mouth in terms of development in the embryo? So here's a look at the phylogenetic tree of all of our invertebrate metazoans. Metazoans just include all of the animals. So here we have at the different branching points are traits or characteristics that we use to end up with all of our different phyla here. So again, body plan is important, whether you have bilateral symmetry, radial symmetry, or pentaradial symmetry. The number of germ layers is important. If you have 2 germ layers, you're called a diploblast. If you have 3, you're called a triploblast. Your body cavity, if you have a body cavity, you're called a coelomic organism. You have a coelom or cavity. And if you don't, you have no coelom, you are acoelomate. And then finally, mouth formation. We have 2 groups, the protostomes and the deuterostomes. And we will definitely get into what each of these mean, but this is just to orient you with how this is displayed on our phylogenetic tree here. So moving on, we have already discussed different planes of symmetry and body plans in previous lectures. So this is just to serve as a reminder for you. I have no idea what happened to this image. That's okay. If you are a bilaterally symmetrical organism, that means you have 2 similar halves. You can divide yourself into a plane that represents a mirror image on the left and the right side. So many of the organisms, the invertebrate organisms that we'll talk about in the coming slides, have bilateral symmetry. They have an anterior end, meaning the end with the head, and the posterior end, meaning the end with the tail. You also, as a bilaterally symmetrical organism, have a ventral side, that is your belly side, and you have a dorsal side, meaning the side that is on your back. And here represented as this lobster is this plane of symmetry going straight between its eyes here. In contrast to a body plan that is bilaterally symmetrical, you can also have radial symmetry. This means that your body plan is arranged in a circle around a central point. We'll talk about this in terms of jellyfish and the sea anemone. So here we have multiple planes of symmetry that we can cut the sea anemone into. It just has to originate from this center point here. So our jellyfish and our sea anemone are characterized by having radial symmetry. There's a special type of radial symmetry that we'll talk about in terms of our echinoderms, and that's called pentaradial symmetry. This is a modified radial symmetry around a central axis, but it occurs in sets of 5s. So here we have an astroidea, here we have an echinoidea. Both are within the phylum of echinoderms, and here you have the central point and 5 points radiating out of this. And finally, we have a body plan where there is no tellable symmetry. This is when you are asymmetric. Our sponges are the characteristic phylum that is characterized by asymmetry. There are no axes of symmetry present within the organism. No matter how you try to slice it or dice it, you can't come up with any type of symmetry. So next, let's move into the process of having a head. This is a very important feature in classifying our invertebrates. We call this cephalization. This is the situation of having a head. It means that you have a collection of organized nervous system, meaning cerebral ganglia, or a brain in more complex organisms. You have a collection of organized nervous system at the animal's anterior end. And the mode of cephalization, having a head, through evolutionary time was promoted through the evolution of bilateral symmetry that allowed the development of an anterior and a posterior end. So when we're talking about the different types of organized nervous systems, we see this represented differently in different invertebrate groups. For example, our insects that we'll talk about in coming slides in this lecture, they have a cerebral ganglion at the front in their head. And looking at a flatworm, they too have a cerebral ganglion at their anterior end, what is technically their head. Next, we need to talk about germ layers. And most animal species undergo a separation of tissues into germ layers during embryonic development. And recall from previous lectures that these germ layers are formed during the process of gastrulation. Each germ layer typically gives rise to a specific type of embryonic tissue and organ. And animals develop either 2 or 3 embryonic layers during their development. If you have 2 germ layers during embryonic development, you are called diploblastic. This means you only have an inner and an outer germ layer, the inner layer being the endoderm and the outer layer being the ectoderm. By contrast, triploblastic organisms have 3 germ layers. This is what we see in our more complex animals, including you and me, but as well as some of the invertebrates we will be talking about. In a triploblastic organism, you have the presence of an endoderm, an ectoderm, but also the addition of a middle layer, a mesoderm. So here we have the endoderm in the triploblastic organism, the ectoderm, and then the presence of this red mesoderm represented here. Digging into further subdivisions of the triploblast, we can begin to talk about the presence or absence of a body cavity. So we can separate animals that may develop an internal body cavity that is derived from that mesoderm. This body cavity is called the coelom. Those with the body cavity are called coelomate organisms. Coelomate invertebrates include the annelid worms, like our earthworms, and those without this cavity are called acoelomate, literally meaning without the coelom. So here we have the true coelomate organisms where, for example, this annelid flatworm, if we do a cross section, this is what the inside looks like. We have the true coelomic cavity. We can break this word down, eucoelomate, meaning U is true and coelomate is to have a body cavity. Now, if you don't have this body cavity, you're called acoelomate. Our acoelomate organisms include the flatworms that we'll talk about in coming slides. A means without here. So acoelomate organisms do not have that body cavity. Again, this is what it would look like in cross section. Notice we do not have that true body cavity as we do with our annelid worm. Now, there are cases in which it may appear superficially that there is a coelom or body cavity, but it's actually not a true coelom. This condition is called pseudocoelomate. Pseudo means false, And so an example of a pseudocoelomate organism is the nematode worm. Again, we can do a cross-section analysis and see that it has something that superficially looks like a coelom or body cavity, but it is a pseudocoelom. And the coelomic cavity is important because it's filled with fluid and it lies between the visceral organs and the body wall. And this is where we have the occurrence of our digestive tract, our urinary tract, our reproductive system, our heart, and our lungs. It also contains major arteries and veins of the circulatory system. So if you ever hear me say that we're nothing but tubes, this is why, because we're coelomate organisms. We have that central body cavity. Okay, so delving deeper again into further subdivisions are bilaterally symmetrical triploblastic eucoelomates. Those are a lot of words, but you can go back to the preceding lecture slides to understand what those mean. So our bilaterally symmetrical triploblastic eucoelomates can be further divided into two groups based upon differences in the origin of their mouth. So when the primitive gut forms, the opening that first connects the gut cavity to the outside of the embryo is called the blastopore. Okay. Most animals have openings at both ends of the gut, the mouth at one end and the anus at the other end. And one of these openings will develop at or near the site of the blastopore. So in our protostomes, meaning our mollusks, our annelids, and our arthropods, the mouth develops first. In contrast with our deuterostomes, the mouth develops second. The deuterostomes include us, members of the chordates or the vertebrates, as well as the echinoderms, our sea stars and our sea fish. Okay, so being a protostome or a deuterostome all has to do with the fate of the blastopore. Does that blastopore develop into a mouth, or does that blastopore develop into an anus? In our mollusks, annelids, and our arthropods, that blastopore develops into a mouth, and in us, as well as the sea stars and their allies, that blastopore develops into an anus. There are other differentiating characteristics between the deuterostomes and the protostomes, namely in the type of cleavage. And cleavage is just the term for the division of cells early in the embryo. So protostomes undergo what we call spiral cleavage. And this means that the cells of one pole of the embryo are rotated and thus are misaligned with respect to the cells on the opposite pole. And this is due to the oblique angle of cleavage relative to the two poles of the embryo. So we have spiral cleavage within our protostomes. In our deuterostomes, we have what's called radial cleavage. And this is where the cleavage axes are either parallel or perpendicular to the polar axis, resulting in parallel alignment of the cells between the poles. You'll also notice here a second distinction called determinate or indeterminate. And this second distinction between the types of cleavage is related to the fate of the blastomeres. And the blastomeres are just those individual cells that are born through cleavage. So in addition to spiral cleavage, our protostomes undergo determinate cleavage. This means that even at an early stage in embryonic development, the developmental fate of each embryonic cell is already determined. So any given cell has a role that it is destined to play in the final individual organism. If you were to remove a blastomere from the growing embryo, it would ultimately result in missing structures or organs that might make the embryo fail to develop into a full individual. In contrast, our deuterostomes have indeterminate cleavage. This means that the fate is not written for any of these blastomeres. Instead, they have the potential to turn into anything that needs to be developed. So it's not committed to a specific cell type early in the cleavage process. And therefore, removal of an individual blastomere does not result in the loss of embryonic structures. So that was a lot to digest. That's all to serve as a preamble to the diversity of the invertebrates that we'll now get into in the next part of this lecture.